Hello everyone. Today's my lecture is on organization of the parasympathetic division. It is also known as craniosacral outflow because the fibers of this division arise from the brain and sacral segments of spinal cord. Parasympathetic division of the autonomic nervous system, that is known as craniosacral outflow because the fibers of this division that arise from the brain or cranium and sacral segments of spinal cord, so that it is known as craniosacral outflow. Cranial outflow or cranial portion of the parasympathetic division arises from the brainstem and it innervates the blood vessels of head and neck and many thoraco-abdominal visceral organs. Cranial outflow that includes the following cranial nerves: 3rd, 7th, 9th and 10th cranial nerves: oculomotor, facial, glossopharyngeal and vagus nerve. Preganglionic fibers of these cranial nerves arise from the neurons that are situated at two different levels: tactile or midbrain outflow and bulbar level or bulbar outflow. That includes 7th, 9th and 10th cranial nerve. Tactile outflow that includes 3rd cranial nerve. Preganglionic fibers are longer and reach the postganglionic neurons, which are situated within the organs or close to the organs innervated by these nerves. So these are the parasympathetic division. Here the preganglionic fibers are long and postganglionic fibers are short as compared to preganglionic neurons. Preganglionic fibers are myelinated but the postganglionic fibers are non-myelinated. Tactile or midbrain outflow. Group of the cells forming Edinger-Westphal nucleus of the 3rd cranial nerve that give rise to tactile fibers. Fibers from this nucleus end in the ciliary ganglion and from the ciliary ganglion postganglionic fibers arises and that supply the sphincter pupillae and ciliary muscles. Bulbar level or bulbar outflow. Preganglionic fibers are the fibers of the 7th, 9th and 10th cranial nerve which arise from the nuclei present in the medulla oblongata. Fibers of the 7th cranial nerve supply the lacrimal, nasal, submaxillary and sublingual glands. Preganglionic fibers of this nerve end in the sphenopalatine ganglion and submaxillary ganglion. This one is a submandibular ganglion. Postganglionic fibers from the sphenopalatine ganglion that supply lacrimal glands and nasal glands. Postganglionic fibers from the submaxillary ganglion that supply sublingual and submaxillary glands. Fibers of the 9th cranial nerve that supply parotid gland. Preganglionic fiber synapse with the neurons of otic ganglion. So this one is the otic ganglion. Postganglionic fibers from the otic ganglion that supply parotid gland. Fibers of the 10th cranial nerve supply the visceral organs of the body. And preganglionic fibers that terminate in the ganglia which are situated on or near the organs. Postganglionic fibers from the ganglia that supply the organs. And vagus nerve supplies almost all the organs that are present in the thorax and abdomen, but not the pelvic organs. Then sacral outflow or sacral portion of the parasympathetic division. Sacral outflow or sacral portion of the parasympathetic division arises from the sacral segments of the spinal cord. It innervates the smooth muscles forming the wall of the viscera and glands such as large intestine, liver, spleen, kidney and bladder and genitalia. Preganglionic fibers arises from the anterior gray horn of second, third and fourth sacral segments of the spinal cord, and it forms a pelvic nerve or nervy regions. These are so preganglionic fibers arises from the second, third and fourth sacral segments of the spinal cord, and it forms a pelvic nerves. Fibers end on the postganglionic neurons, which are situated on or near the visceral organs. Fibers from the postganglionic neurons supply descending colon, rectum, urinary bladder, internal sphincter, urethra and accessory sex organs. Sacral parasympathetic fibers supply those visceral organs which are not supplied by vagus nerve. Now most of the visceral organs have a dual innervation. For example, are supplied by both the sympathetic and parasympathetic division of autonomic nervous system. The two divisions that produce antagonistic effect on each other and provide a very fine degree of the control over the effector organ. Some of the visceral organs are innervated by one division of the autonomic nervous system only. For example, uterus, adrenal medulla and most of the arterioles are innervated by the sympathetic division only. And the glands of the stomach and pancreas that are innervated by the parasympathetic division only. In case of the sphincter's muscle, both adrenergic and cholinergic innervation are excitatory, but one supplies the constrictor component of the sphincter and other one supply the dilator component of the sphincter. Almost all the visceral organs are supplied by both sympathetic and parasympathetic division of autonomic nervous system, and the two divisions produce antagonistic effect on each organ. When the fibers of the one division supplying to an organ that is sectioned or affected by lesion, the effect of the fibers from the other division on the organ become more prominent. Then autonomic neurotransmitters, parasympathetic fibers, preganglionic fibers that releases acetylcholine and postganglionic fibers that release acetylcholine, and sympathetic fibers that includes preganglionic fibers that release acetylcholine and postganglionic fibers that norepinephrine mainly or epinephrine also. All the preganglionic fibers, sympathetic as well as parasympathetic, release the acetylcholine. All the postganglionic parasympathetic fibers release acetylcholine. Most postganglionic sympathetic adrenergic fibers release norepinephrine. A few postganglionic sympathetic or cholinergic fibers release acetylcholine. So cholinergic fibers. The postganglionic sympathetic cholinergic nerve fibers supplying the sweat glands, blood vessels in the heart and skeletal muscles. Now about the acetylcholine. That is a cholinergic neurotransmitter and it possesses excitatory function. It produces the excitatory function by opening the ligand-gated sodium channel. What is the source of acetylcholine? So acetylcholine, it is the transmitter substance at the neuromuscular junction and synapse. And it is also released by the following nerve endings: preganglionic parasympathetic nerve, postganglionic parasympathetic nerve, preganglionic sympathetic nerve, and postganglionic sympathetic cholinergic nerves. Nerves supplying eccrine sweat glands, sympathetic vasodilator nerves in the skeletal muscle, then nerves in the amacrine cells of the retina, and many regions of the brain. So cholinergic neurons, neurons which secrete acetylcholine at their nerve endings. Examples include all the preganglionic, autonomic, parasympathetic as well as sympathetic endings, postganglionic parasympathetic endings, postganglionic sympathetic endings which innervate the sweat glands, skeletal muscle, blood vessels, means sympathetic vasodilator nerves, neuromuscular junction, many parts of the brain, especially the cerebral cortex, thalamus and forebrain nuclei, and endings of the some amacrine cells present in the retina. Then synthesis of acetylcholine. So acetylcholine is synthesized in the cholinergic nerve endings. Synthesis takes place in the exoplasm and acetylcholine is stored in the vesicles. It is synthesized from the acetyl coenzyme A. It combines with the choline in the presence of the enzyme choline acetyl transferase to form the acetylcholine. So acetyl coenzyme A combines with the choline. The presence of the acetylcholine acetyl transferase, it forms acetylcholine. Acetylcholine is divided into choline and acetate in the presence of the acetylcholine esterase. Action of the acetylcholine is short-lived. Within the 1 millisecond after the release from the vesicles, it is hydrolyzed into acetate and choline by the enzyme acetylcholine esterase, and this enzyme is present in the basal lamina of the synaptic cleft. Effect of the acetylcholine of localized cholinergic discharge are generally discrete and short lasting because the acetylcholine is rapidly removed from the nerve endings due to high concentration of the acetylcholine esterase at cholinergic nerve endings. Adrenal medullary hormones are the amines derived from the catechol, and so these hormones are known as catecholamines. Catecholamines secreted by the adrenal medulla. Adrenaline or epinephrine, noradrenaline or norepinephrine, and dopamine. Synthesis of catecholamines. Catecholamines are synthesized from the amino acid tyrosine in the chromaffin cells of adrenal medulla. These hormones are formed from the phenylalanine also, but the phenylalanine has to be converted into tyrosine. So stages of the synthesis of catecholamines. First of all, formation of tyrosine from phenylalanine in the presence of phenylalanine hydroxylase. Then second step is uptake of tyrosine from the blood into the chromaffin cells of the adrenal medulla by active transport. Then conversion of tyrosine into dopamine. or DOPA, dihydroxyphenylalanine, by hydroxylation in the presence of tyrosine hydroxylase. Decarboxylation of DOPA into dopamine by DOPA decarboxylase. Entry of the dopamine into granules of the chromaffin cells. Hydroxylation of the dopamine into noradrenaline by the enzyme dopamine beta hydroxylase. Release of the noradrenaline from the granules into the cytoplasm. Methylation of the noradrenaline into adrenaline by the most important enzyme, that is known as phenylethanolamine N-methyl transferase, PNMT, and PNMT that is present in chromaffin cells. Metabolism of the catecholamines. 85% of the noradrenaline is taken up by sympathetic adrenergic neurons, and remaining 15% of the noradrenaline and adrenaline are degraded. Stages of the metabolism of the catecholamines. Methoxylation of the adrenaline into meta-adrenaline and noradrenaline into meta-noradrenaline in the presence of catechol O-methyl transferase, COMT. Meta-adrenaline and meta-noradrenaline are together called metanephrines. Oxidation of the metanephrines into vinyl mandelic acid VMA by monoamine oxidase. Removal of the catecholamines. So catecholamines are removed from the body through the urine in three forms: 15% as a free adrenaline and free noradrenaline, 50% as a free or conjugated meta-adrenaline and meta-noradrenaline, 35% as a vinyl mandelic acid VMA. Effect of the norepinephrine are more pronounced than acetylcholine as it spreads further. In the blood, the epinephrine and dopamine come from the adrenal medulla, while norepinephrine diffuses from the adrenergic nerve endings. So, adrenergic neurons. So, neurons which secrete norepinephrine or epinephrine at their endings. Examples include postganglionic sympathetic endings other than above: cerebral cortex, hypothalamus, cerebellum, brainstem, spinal cord, and adrenal medulla. Some autonomic fibers release neuropeptide besides the classical neurotransmitters, and these include adenosine triphosphate. The small vesicles of the postganglionic noradrenergic fibers contain ATP. The low frequency stimulation causes ATP release. Second one is galenin vasoactive intestinal peptide, calcitonin gene related peptide or substance P, that are released along with the acetylcholine on the sympathetic stimulation to sweat glands. Third one is neuropeptide Y. It is another transmitter present in the large vesicles of the postganglionic sympathetic endings that supplies to the vasculature of the viscera, skin and skeletal muscles, and that releases on high frequency stimulation in addition to norepinephrine. This is the list of the reference books. Thank you very much.